Hello everyone, welcome back to my channel. Thank you for tuning in for another exciting and informative video today. So today we are going to talk about analyzing single cell ATAC-Seq data, and in this video I'm going to talk about what is ATAC-Seq, the difference between bulk and single cell ATAC-Seq, the analysis and pre-processing steps for ATAC-Seq, and finally we'll be demonstrating how to analyze such a data set in R. So ATAC-seq stands for Assay for Transposase Accessible Chromatin with Sequencing. It's a molecular biology technique used to analyze chromatin accessibility. Chromatin accessibility refers to the ease with which proteins like transcription factor can bind to the DNA. So just a quick recap, the way on how DNA is organized in a cell. So DNA is wrapped around certain proteins. So there are histone proteins which come together and form a unit called a nucleosome, and DNA is bound around this nucleosome. So this wrapping of DNA around histones helps in packaging and condensing the DNA strand. And these nucleosomes further coil into higher order structures, ultimately giving rise to chromosomes. But you must be wondering at this point as to what is the use of studying this open chromatin region. How can it help us? So this open chromatin, so the chromatin accessibility, or the opening and closing or the reach of these regions will basically give us an idea on which genes are being transcribed. So when the chromatin is open, when it is not bound by nucleosomes or histones, these regions are accessible for the DNA binding proteins and transcription factors and regulatory elements to bind to these regions and enables transcription. So basically, it can give us an idea as to which regions are being actively transcribed by looking at which regions are open, basically have chromatin accessibility by this method, that is ATAC-seq. So ATAC-seq works by using a transposase enzyme, a TN5 transposase enzyme, which tags these accessible regions of chromatins with sequencing adapters. So these tagged regions are then sequenced to identify which DNA is accessible and open for regulatory protein binding. This helps in understanding the gene regulation by revealing the landscape of chromatin accessibility across the genome. So by identifying regions of open chromatin, it helps in understanding the regulatory networks that govern the gene expression in different cell types and conditions. So let's say if you have two conditions and if they have different regulatory or open chromatin accessibility regions, it can give us an idea on which genes are being transcribed in one condition versus the other. It's a powerful tool for studying development, disease mechanisms, and identifying potential therapeutic targets. You might have noticed in a lot of papers where they mention ATAC-seq data, you might have seen such peaks or such graphs where you see there are peaks being shown at certain genomic locations. So in the upcoming slides, we'll be talking about the workflow steps to analyze the ATAC-seq data, but this is how essentially the ATAC-seq data is represented and interpreted, as to these regions of peaks are interpreted as a higher signal, or basically a signal of open chromatin in that region. So just like the difference between the bulk RNA and the single cell RNA-seq, similarly with bulk ATAC-seq, the chromatin signal is averaged across all the cells in the sample, which limits the insights into the chromatin accessibility of specific cell types in the data. Whereas for the single cell, the chromatin accessibility for each cell type can be visualized or interrogated. So basically, the bulk ATAC-seq is useful more for identifying the different chromatin accessibility between experimental conditions or disease or tissue types. Whereas for the single cell, it's more to identify the cellular heterogeneity of the gene expression, going back to which regions are more accessible for gene expression between rare cell types, characterizing cell lineages, trajectories, and understanding the regulatory dynamics at the single cell level. So as I said, the single cell ATAC sequencing can help identify cell type and classification by understanding the cellular heterogeneity and lineage relationships. It can also help in identification of regulatory elements and can provide insights into the regulatory networks. It can also provide insights into processes like cell differentiation, reprogramming in response to environmental stimuli or disease. It can be used to perform transcription factor binding analysis, understanding what are the transcription factor binding in different cells, whether we do see any differences in the transcription factor binding, or preferences between any specific cell types in any conditions or when comparing between conditions. We can also understand epigenetic heterogeneity within cell populations, impacting the gene expression. We can also understand disease mechanisms and discover biomarkers. It can also be useful in drug discovery and development, which helps us in understanding drug mechanisms of action and can help for us to predict the drug responses and identify potential drug targets. In addition to that, it enables high throughput screening of regulatory elements and non coding variants for their effects on gene regulation and cellular phenotypes. So, this is the single cell ATAC-Seq workflow, which can broadly be divided into pre processing and downstream analysis. Much like how we have for single cell RNA-seq. So we start with the reads from the sequencer, and if the samples are multiplexed, then the first step we do is to demultiplex the reads. And if you're not sure about demultiplexing, in one of my previous videos, I've explained that. So I will be adding the link to that video in the description section below. So once the reads are demultiplexed, we perform adapter trimming, getting rid of adapters. We perform some additional quality control. We perform the alignment, and we perform post alignment QC to make sure that we retain high quality alignments. All of these steps are pretty much standard for any high throughput sequencing analysis pipeline. Followed by that, we perform something called as peak calling and annotation. So peaks, calling the peaks are basically identifying regions of open chromatin. So peaks would give us an idea on which regions have open chromatin, and these regions can also be annotated as to what is the closest gene, or in which region within the gene body it's present in, whether it's in the intronic region, in the promoter region, at the transcription start site. All of that can be annotated as well. And once the peaks are called, we generate something called as a cell by feature matrix. You might have heard me talking about cell by feature matrix for the single cell RNA-seq data, where in single cell RNA-seq, we have the rows as the features, which are genes, and the columns as the cell. Similarly, a matrix here is also generated, but instead of the rows being genes, we have the rows as the regions of open chromatin. If you're not clear about that, while I demonstrate, I will be showing you how a cell by feature matrix would look like for single cell ATAC-seq. Once we have the cell by feature matrix, we identify the features which are capturing the majority of the variation, which can be used for performing further dimensionality reduction to cluster the cells by similarity in terms of the similarity in certain features. And then we perform cell type identification. This is much more analogous to what we do for single cell RNA, and this will be more clearer as we go into each of these steps while I demonstrate. In addition to that, if we find any batches, if we have data from different batches, we would also make sure that we correct for those batches before we go on to the downstream analysis. In the downstream analysis section, in addition to the cell identity annotation, we can also perform differential accessibility analysis, where we can investigate the differential accessible regions in certain cell types across conditions. We can perform trajectory analysis. We can also perform co-accessibility analysis. In addition to that, we can also integrate different types of omics data and generate further insights. Like, it is common to integrate single cell ATAC with the single cell RNA, which can give us additional evidence in terms of the ATAC giving us an idea on the regions of open chromatin, and the gene expression giving us further evidence of those genes in those specific regions being expressed. And these can be compared, and this can be compared between different cell types across conditions. However, in today's demonstration, we are going to focus on the pre-processing aspect of this workflow, where we are going to use the cell by feature matrix and we are going to perform a bunch of steps until we perform clustering. So there are a lot of packages and tools available to process single cell ATAC-Seq data in R and Python. This tabular compilation is from a Nature article published in 2021, which basically compares various R packages to process single cell ATAC-Seq data and perform a bunch of things that going from the most comprehensive to the least comprehensive. There are also certain Nextflow pipelines and packages in Python that can also be used to process this data. So for the demonstration today, we are going to use Signac to pre-process our single cell ATAC-Seq data. And the data that we are going to use for today's demonstration is human peripheral blood mononuclear cells provided by 10x Genomics. And these are our requirements today. So in addition to Signac, we'll require Seurat, Tidyverse, and Ensemble database package. This package essentially will help us to add some annotations to our data. And this will make more sense once I go over the demonstration. So for today's demonstration, we will be following this vignette that is provided by Signac on the website to analyze the PBMCs. We will be requiring these files. So we have dealt with the raw data and the metadata when we were dealing with the single cell RNA-seq. The raw data here is a little different and we will be taking a look at that when I go through the demonstration. But in addition to the raw data and the metadata, we also have fragments file here. And again, we will be talking a little bit more about this when I'm going through the demonstration. But these are the files that you would essentially have to download and you will require for this tutorial. They've also provided the download code here, which you can run to download all the files. And you will be finding the link to this vignette in the description section below. So we will first start by loading the libraries and ensure that you have the packages required for these tutorials installed. While I was running through this, while I was preparing for this demonstration, I encountered certain errors which required me to install certain dependencies. So I've added these the codes here to install specific libraries and dependencies so you wouldn't have the same errors. So make sure you install these libraries and dependencies before you load your libraries and run your code. Now that the library is loaded, I just want to quickly show you that I have all my data downloaded here as well. So these are the fragment of, this is the fragment file, this is the fragment index file, this is the raw data, and this is the metadata. Since the fragment file is something that we have not taken a look at when we were dealing with the single cell RNA-seq, let us take a look at what is this fragment file. So I'm just going to read this data in. And I'm just going to read the first 10 rows. And let us take a look at this file. So this fragment file looks like some sort of a bed file where the first three columns are the genomic locations. Then it appears that we have a cell barcode and there is a number. So let us go back to the documentation to figure out what that is. So on the 10x Genomics platform, they have provided details of how this fragment file is generated, which is an output of the Cell Ranger ATAC count pipeline. So they have provided the details of this file. So essentially, this file, the first three columns are the genomic locations, the chromosome start and the end. The fourth column, as I said, is a cell barcode, the 10x cell barcode, and the last column holds the total number of reads associated with that fragment. So it's essentially the number of reads that support that fragment. Coming back to the code, now we can start reading in the data. So the first thing we'll do is we'll read the raw data, that is the feature, the cell by feature matrix. So we'll read, since this is a .h5 file, we can read it as d10x h5, and we provide the file name with the path. Now that this data has been read, let us take a look at what this counts cell by feature matrix looks like. So here it's At a first glance, this is a sparse matrix. Only non-zero values are stored here. This is very much like the counts data that we have for the single cell RNA-seq, but the major difference is that the feature here are not genes, but these are essentially the regions of the peaks that are predicted to be open chromatin by the peak callers. So basically, each region here is predicted to represent the region of open chromatin, and the numbers here are the number of TN5 integration site in each of the cell that maps to that region. Now, using the counts cell by feature matrix and the fragment file, let us create a chromatin assay. The min cells parameter ensures to include features that are detected in at least these many cells, and min features parameter ensures to include only those cells that has at least these many features detected. These are the numbers or the thresholds that have been given in the standard vignette, and I'm just following that. So let us run this. So now this chrome assay has been created, a chromatin assay has been created. Let us take a look at that. So essentially, it's like an object with bunch of slots, saving or storing all the information associated with the chromatin assay, including the chromatin, the chromosome names, the regions of these chromatin accessible regions, the genomic locations of that, the strand, the information on the chromosomes, metadata, if there is any motifs, fragment information, annotations could be added, and there is a bunch of additional information associated with the chromatin assay. So now that we have created a chromatin assay, let us read the metadata. So we've also been given the metadata associated with this data set. So let us create a metadata object. Since this is a CSV, let us also take a look at this metadata. So this metadata file reports a bunch of the reads that are the total number of reads in each of the cells, the duplicate, the chimeric, the unmapped, the low map quality reads, reads mapping to the mitochondrial regions, those that pass the filters, those are that are in the TSS regions, in the enhancer region, in the promoter region, in the blacklist region, and these numbers or this metadata for the 10x data sets is provided as an output from Cell Ranger. But in the vignette, they mentioned that if it's a non-10x data set, that these can be calculated using the Signac package, and they have instructions on how to do that. And a bunch of these values or these values from these columns would be utilized for some of the QC that we will be performing next. So now that we have our chromatin assay and our metadata, let us create a SeuratObject using these. For the counts, we will provide the chrome assay. The metadata is in the metadata object, and let us name this assay as ATAC. And let's store this in another object called PBMC. Now taking a look at this Seurat object, This will be familiar because we have taken a look at a lot of Seurat objects in the past. So this is just another Seurat object with ATAC as one of the assays. So we can have multiple assays in Seurat object. We can also have RNA in addition to the ATAC, but here we are just dealing with one type of omics data set. So we just have one slot, which is one within the assay slot. We just have one assay, that is ATAC, and it has all the information associated with the ATAC-Seq experiment. A final step before we move on to the quality control is to add gene annotations to our Seurat object. This can be useful for the downstream functions to pull the gene annotation information from the object. So let me quickly show you where this information will be stored within the Seurat object. So within the assays, we have ATAC-Seq, and within the ATAC-Seq we have an annotation slot. So right now this slot will be null because we don't have any annotations added in, but we hope to add annotations so that we can have all the information in one object and it can be pulled when required. So we are going to pull the annotation from the ensemble database using the function get G ranges from ensemble database. This function essentially extracts all the transcript information on all chromosomes from the ensemble database. And the ensemble database is the one that we loaded as a library before. So this is the ensemble database that we will be providing, and let us store this in an object called annotations. So let us take a look at the annotations object now. So this annotation object extracts the exon and the coding region information from the transcripts across the genes across all the chromosomes along with the strand and the genomic location. If you notice that here the chromosome, the sequence names are essentially the chromosomes, but they are missing the prefix CHR. So these things can cause conflict. And again, our data has been mapped to the HG19 from UCSC. So it's important that we add the CHR prefix here to the chromosomes to avoid any conflicts downstream. So let us first change that in the object, in the annotations object, and then add it to the Seurat object. So let me show you how we do that. So the sequence levels of our annotation annotations object is essentially these. So this is what I was saying, that all the chromosomes here are missing the CHR prefix. So we just paste that. Let us run this again to make sure that we have the CHR prefix added. And now let us add this annotations object to our Seurat object. Now let us quickly check again if the annotations have been added. So now you see that this slot was null, and after adding the annotations, this has been populated with the annotations. So now that we have all the data together in a Seurat object, the next step is to perform quality control and to filter poor quality cells. So there are certain metrics that we need to calculate and we need to look at that would help us assess the quality of the data and filter the poor quality cells. It's important for us to understand what these quality metrics tell us about the data, and it's important that we have a good theoretical understanding of these metrics before we jump onto doing it, demonstrating it, and performing it practically. So let us go back to the presentation to understand what they mean, and we'll come back and continue the demonstration once we've understood the quality control metrics that we will be taking a look at. So in order to assess the quality of the single cell ATAC-seq data, we will be looking at these 5 metrics. So we will be looking at the nucleosome banding pattern, the transcriptional start site enrichment score, the total number of fragments in the peaks, the fraction of fragments in the peaks, and the ratio of reads and genomic blackest regions. So quick recap on chromatin organization. So two of each of the histones, H2A, H2B, H3 and H4, come together to form a histone octamer which binds and wraps approximately 146 base pair of the DNA. So these nucleosomes are joined together by a DNA that runs between them, which is called as a linker DNA. Now, the amount of DNA that is per nucleosome that is bound by the nucleosome can be determined by treating the chromatin with an enzyme that cuts the DNA. Now, one such enzyme has a specialty to preferentially cut the linker DNA between the nucleosomes well before it cuts the DNA that is wrapped around the octamers. So, by regulating the amount of cutting that occurs after the application of such an enzyme, it is possible to stop the reaction before every linker DNA has been clipped. So at this point, the treated chromatin will consist of mononucleosomes, dinucleosomes connected by the linker DNA, trinucleosomes, and even some of the linker DNA that is not bound by any nucleosomes. Now, if we think about the goal of ATAC-Seq, the goal of the ATAC-Seq experiment is essentially to identify the regions of DNA that are not bound by the nucleosomes, which indicates that the genes in those regions are being actively expressed or regulated. So essentially, we will be wanting to retain only those fragments that are not bound by the mononucleosomes or dinucleosomes, which are essentially free of nucleosomes and which are open and accessible for the other DNA binding proteins to bind to these DNA. So in the perspective of single cell ATAC-seq, we would essentially want to retain those cells that have higher number of fragments that are not bound by nucleosome. So we measure that by calculating nucleosome signal. So nucleosome signal is a ratio of the fragments bound by mononucleosome over nucleosome free fragments. So the cells which have lower nucleosome signal indicates that it has enrichment of fragments that are not bound by nucleosomes. And in addition to the nucleosome signal, we can also visualize the nucleosome banding pattern for the cells with low nucleosome signal versus the cells with high nucleosome signal. So successful ATAC-seq experiment should generate a fragment size distribution with decreasing and periodical peaks corresponding to nucleosome free regions and followed by the mononucleosome, dinucleosome and trinucleosome regions. So intuitively, if our cell has a high number of nucleosome free fragments, the nucleosome free fragments would be of a smaller length. So we should expect to see a high frequency of these fragments in our cells with low nucleosome signal, followed by the fragments that go up increasing in size, which indicates that they might be bound around mononucleosome, dinucleosome, and trinucleosomes. We can compare this banding pattern with the banding pattern observed in cells with higher nucleosome signal, as we can see that they have different banding pattern indicating higher lengths of these DNA fragments, indicating that these must be bound around the mono or the dinucleosomes, and essentially these are the type of this is the banding pattern that we would expect to see, and these are the cells that we would essentially want to retain. So the next metric that we will be taking a look at is the transcriptional start site enrichment score. So the fragments from the nucleosome free regions are expected to be enriched around the transcription start site. The idea here is that if these fragments are from the open chromatin region that is not bound by any nucleosome, the genes in this region will be actively transcribed or regulated, and hence the nucleosome free regions should be expected to be enriched around the transcription start site of these genes. So the transcription start site enrichment score is calculated by taking a ratio of the fragments centered at the transcription start site over the fragments in the TSS flanking regions, that is around the TSS regions. So in with regards to the single cell ATAC, we ideally want to retain all the cells that have a high transcription start site enrichment score. The third metric that we look at is the total number of fragments and peaks. This is a measure of cellular depth or sequencing depth or complexity. Cells with very few reads may need to be excluded due to low sequencing depth, and cells with extremely high levels may represent doublets, nucleic clamps, or other artifacts. This can be seen equivalent to the number of counts, transcript counts in single cell RNA-seq. Next, we also look at the fraction of fragments and peaks, and this represents the fraction of all the fragments that fall within the ATAC-seq peaks. Cells with low values, typically less than 15 to 20%, represent low quality cells, or technical artifacts that should be removed. And lastly, we look at the ratio of the reads in genomic blacklist regions. So the ENCODE project has provided a list of blacklisted regions, and these are the regions in the genome that have high unstructured anonymous signals in next generation sequencing experiments. These regions can be problematic for high throughput sequencing aligners, and they may be caused by repetitive elements or other anomalies. So cells with a high proportion of reads mapping to these regions can represent technical artifacts and should be removed. So all these three metrics can be obtained from the output of the cell ranger. However, this can also be calculated for the non-10x datasets using Signac. Now that we understand what these quality control metrics are, let us calculate those. So we first start by calculating the nucleosome signal, and let us store it back into the object. I'll show you what these values look like in a bit, but let us first compute all the QC. So next we calculate the TSS enrichment. The TSS enrichment score has been calculated, and this can take a while to run. So now this has finished running. Let us also calculate the ratio of the fragments in the blacklisted region as well as the fraction of reads in the peak. So we will be utilizing some of the columns from the metadata. And we save this into another variable. And we also calculate the fraction of reads in the peaks. So for that, and we save this in. Now that all the QC have been calculated, let us view the metadata. So as you can see that in addition to the metadata columns, we have the nucleosome signal calculated for each of the cell. We have the TSS enrichment, we have the nucleosome percentile, TSS percentile, the blacklist ratio, and the percentage of the reads and the peaks. So now that we have calculated all of these QC, let us visualize some of these metrics so that we can come up with the cutoffs, and I'll quickly show you how we do that. So there is a function called density scatter which allows us to visualize different metrics and come up with the thresholds. So let me get all the metrics that we have in our metadata. So let us plot the counts versus the TSS signal enrichment, the TSS enrichment score. And we'll set the quantiles as true, and you will understand why that is. So when I run this, I should end up with a plot that looks like this. So density scatter generates a scatter plot like this, which allows you to visualize data for two of your metrics from your metadata. So in this case, we have plotted the number of counts on the x-axis and the TSS enrichment score on the y-axis, and each cell is colored by the density of points. The red lines that you see going across the plot, so the horizontal lines are, the vertical lines are the quantiles, 5th quantile, 10th quantile, 90th, and the 95th quantile for the n counts. The corresponding values for these quantiles can be found at the top of the plot, and the horizontal red lines that you observe are similarly these quantiles for the TSS enrichment scores. So visualizing data in this way can help you come up with your filtering thresholds to ensure that you're not choosing a threshold that's too stringent that majority of your cells are filtered out, but at the same time choosing a threshold that retains high quality of cells. Similarly, we can create another density scatter plot with another metric. So in this case, we will be plotting the nucleosome signal, and let's do the TSS enrichment again. And let's set the log of x-axis as true, and let's show the quantiles. And I'm saving these plots into different variables so we can visualize them together. So this is the second density scatter plot that we created, plotting the nucleosome signal and the TSS enrichment. Again, this can be very helpful in choosing what nucleosome signal threshold that we should be choosing to keep our cells having, to ensure that our cells are of high quality, having the low nucleosome signal, but at the same time having a high TSS enrichment. So similarly, you can create similar plots for all the metrics that you want to check, and then come up with thresholds for each of those metrics to filter out poor quality cells from your data. Just like how we created a violin plot for our QC metrics with the single cell RNA-seq, let us create a similar violin plot for all the metrics that are of interest to us with our single cell ATAC-seq data. These are all the columns that are present in our metadata. So I'm just choosing the metrics that I want to visualize. So these violin plots show the distribution of your cells in terms of the number of counts, number of features, the TSS enrichment score for your cells, the nucleosome signal, the blacklist ratio, and the percentage of the reads and peaks in your cells. So someone recently asked me how could one decide on the thresholds based on these violin plots. So to give you an example, let's say for the TSS enrichment, ideally we would want to keep cells having high TSS enrichment score. So looking at this plot, if I were to choose a threshold 7.5, I would be certain that I'm losing majority of my cells because that threshold is way too stringent. Let's say if I decide on a threshold of 1 or maybe 2, then I know that the cells that I will be retaining will have some of the cells having lower TSS enrichment score. So good TSS enrichment score would be anywhere over 3 or 4 that will retain majority of my cells, at the same time ensure that my cells have good TSS enrichment scores. So these plots can be helpful. These visualizations are helpful for you to come up with your thresholds that are not too stringent, but at the same time ensures that you retain a majority of the cells and those are of acceptable quality. Now that we have calculated our QC and visualized and come up with the thresholds, let us go ahead and subset our data. And here we start providing all the metrics and the thresholds for them. So finally, we retain all the cells with greater than 3000 and less than 30,000 counts, having greater than 15% of the reads in the peaks, having less than 5% of the reads mapping to the blacklisted regions, have the cells having less than 4 nucleosome signal value, and having greater than 3 enrichment TSS enrichment score. So after filtering for the poor quality cells, next we perform normalization. So for normalization, we run this function. This essentially normalizes for differences in sequencing, cellular sequencing depth across the cells and also across the peaks. So let us run this. Next, we find the variable features, and for to find the top features, we use the function find top features, and here you can set the minimum cutoff, but I'm going to set the quantile 0 because I want to consider all the peaks instead of top 25% or top 75%. And I'm going to run this, and then we perform dimensionality reduction using the top features identified. This can be seen as equivalent to performing PCA with the single cell RNA-seq data. I'm just going to add the comments here. The first component of the reduced dimension representation often captures the technical variation, that is the sequencing depth rather than the biological variation, and this can be evaluated with a function called depth correlation, which basically correlates the sequencing depth with the reduced dimension. So it generates a plot that looks like this. So the first component here shows a very high correlation with the sequencing depth, indicating that it's capturing the sequencing depth, the technical variation, and not the biological variation. So we will proceed with the following steps with the rest of the dimensions or the rest of the components, except excluding the first component. Now that we have our data in a low dimensional space, let us proceed with performing UMAP, finding the neighbors and finding the clusters. These are the standard steps that we perform for our single cell RNA-seq as well. So we perform non-linear dimensionality reduction for visualization, and we find nearest neighbors and perform and use them for clustering. So let us go ahead and perform UMAP. And as I said previously, we will be excluding the first component as it is capturing the technical variation. So we will be running for dimensions 2 to 30. Let's run this. Followed by running UMAP, let us find neighbors. And this will be used for clustering. And finally, let us find clusters. So find clusters function allow us to choose between different clustering algorithms. This is what they use in the vignette. So I'm following the same one. And finally, let us visualize our data. So after pre-processing our single cell ATAC-seq data, it looks something like this after performing clustering. These are the several clusters that are present in our single cell ATAC-seq data. And this is where the pre-processing of our single cell ATAC-seq ends. Followed by this, there are several downstream analysis that can be performed. A common approach is to integrate with the RNA single cell RNA-seq data, as well as performing differential accessibility between these cells to compare the different chromatin accessibility regions between cells of interest. So that's all I had for this video. I hope you found this video informative. I will be adding the links to several resources as well as the vignette that I followed in the description section below. I will also upload my script to the GitHub repository and link that GitHub repository in the description section as well. If you enjoyed this video, please consider giving it a thumbs up. Don't forget to subscribe to my channel for more bioinformatics related content. If you have any questions or suggestions for new topics, please leave it in the comment section below. Share this video with your friends and co-workers if you found it helpful. I appreciate your support and I'll see you in the next video.